Hey guys, welcome to the video on Chapter 8, Lesson number 2. And as always, start with the opening questions. So you're going to want to go ahead and look at these questions, pause the video, take a few minutes to try to answer them, and then come back and we'll go over the answers. So go ahead and pause the video now. Okay, so the answers, guys. The first part, which organelle is involved in photosynthesis? This is a revision from from Chapter 6. The answer is it's the chloroplast. That's the organelle that we're going to be talking about mostly today. Next question is, which organelle is involved in the storage of materials? If you said vacuole here, you would have been correct. This is another important organelle we're going to talk about today. And the last part is, what is metabolism? Metabolism is just the process of breaking down and building up molecules within the body. And there you go. So, lesson 2, guys. I have, again, 3 objectives for you guys. First off, I want you guys to be able to summarize the 2 steps in photosynthesis. There's 2 broad steps that happens within photosynthesis you're going to have to learn about today. Secondly, I want students to be able to explain the function of chloroplasts in photosynthesis. So I want you guys to know what the chloroplast does in photosynthesis and why it's important. Third one is I want you guys to be able to describe and diagram the electron transport chain. This one's going to be a little bit of a challenge for a lot of students, so don't feel bad if it takes some effort. I'm going to have about this video and my own video on TED Ed that's going to talk about, not my video, it's someone else's video, but another video on TED Ed that's going to really go over this idea, and I'm hopefully going to help you guys understand what's happening with photosynthesis and electron transport chain. Okay, quick review on photosynthesis. You take carbon dioxide, water, and sunlight, turn it into sugar. Sugar is this molecule right here. And this right here, if you might recognize it, is oxygen, something you might need to survive for breathing. It's good. I like to breathe. Photosynthesis has 2 phases to it. The first phase is called the light-dependent reactions. Now, if you know your English, you know that the word dependent means you have to have it. So first phase has to have light in order for it to work. However, the second phase is called the light independent reactions. And this phase right here is telling you that you don't have to have light to make this process work. And that'll be actually really important in some plants because some plants don't want to run these reactions during the daytime. You'll find out why later on. So, phase 1, the light-dependent reactions. Well, the chloroplast. This is the organelle right here that is responsible for all of photosynthesis. And it basically looks like this little green thing with these weird little pancake-looking objects that appear on there. And you have these other things called the granulum and the stoma. We're going to learn what all these mean today and kind of what they do. The first part's called the thylakoid. The thylakoid is where the light-dependent reactions take place. The thylakoids are these little disc-shaped things. If you look carefully at the image, you see how there's these small little discs that take place right here. These are all the light-dependent reactions take place. You see how many of these little discs there are. There are quite a few of them. And those are just called thylakoids. The second one is called a grana. It goes, a grania or a granum, if you want to have it for plural, is just a stack of thylakoids. So you see how these are kind of stacked into like a bunch of coins stacked on top of each other? A stack of thylakoids is just a granum. The last part is called a stoma. This is the fluid spilled space outside of thylakoids. And this is where the light-independent reactions take place. So light-dependent happen here, and the stoma is where everything else has. So the stoma, you see all this blank space in between where you have all these, you see all these thylakoids and all these granum, but there's space in between them. This space in between them is called the stroma. And this is just where the light independent reactions are going to take place. So, pigments. Pigments are the light-absorbing molecules that you're going to find inside of the thylakoids. And each one is different. And you should probably notice each plant has a different color. And the reason why each plant can have different colors is because it uses a different style of pigment. The most common pigments out there are called chlorophyll A and chlorophyll B. And they absorb different color spectrums, different parts of the color. Now this chart right here is a good example of what absorbs what. And let's go ahead and look at the white dotted line for chlorophyll A right here. Chlorophyll A absorbs a good section of the blue part of the spectrum, but when it gets down towards the green part of the spectrum right here, it stops absorbing anything. So chlorophyll A really only absorbs around blue and purple, but it does not absorb green, which is why plants are ironically green. But as it goes on here, the second it gets towards the red part of the spectrum, the red-orange, all of a sudden it begins to absorb more of the light. So chlorophyll A is able to absorb both blue and orange and red, but it can't really do the yellows and the greens, which is why a lot of plants have a yellow and a green pigment to them, color to them, is because chlorophyll A cannot absorb those particular colors. Now, the next one is chlorophyll B, which is the green one here. As you see, chlorophyll B is a lot better at absorbing the blue light. But as you can tell, it's really only good at absorbing the blue. After it absorbs blue, it's done. It can't do anything else. It doesn't absorb anything else on the spectrum. So chlorophyll B just only absorbs this middle section right here. And the last one, called a carotenoid, which I believe is the stuff you see inside of carrots and things like that, if I remember right, is they absorb a little closer along the spectrum. They can do more blues and some of the green part of the spectrum, but they also can do some, but they're not really as good. They're almost a lot like the chlorophyll A, except that it does a little bit better in this region here, and it absorbs a different color spectrum. So each one of these pigments can absorb a different spectrum of wavelength of the colors that we have, and that gives them a different ability to absorb energy. So, the electron transport chain. This right here is part of the light-dependent reactions, and you're probably looking at this thinking to myself, oh my Lord, looks complicated. And in a sense, yeah, it basically is, but I'm not going to require that you guys know every single step along this little chart right here. Essentially, here's how photosynthesis works within the light-dependent reactions. The first thing that happens is that light is absorbed by this first protein right here. This is one of the first pigments that's going to be coming into the system. And what happens is this light is used to split a water molecule into its component parts. So it brings it into hydrogen and into oxygen. And it steals one of the electrons. So this electron then, right here, gets moved along through photosynthesis, and it reaches another protein right here, where light then again hits this protein, and it changes this electron and allows it to move farther along the system. And as it does that, what happens is that on the outside of the membrane here, out in the stoma, a hydrogen molecule and a molecule called NAD combines with a hydrogen molecule plus this electron, and you get NADPH. Now, NADPH is going to be important later because NADPH is going to be used in the creation of sugar later on. So let's think of this as kind of like an intermediate. His job is to now move someplace else and help to build a sugar molecule. Now, what happens next is that if you looked over here, you see that as these waters are being split, a lot of hydrogen molecules are being left around. That's going to make this thing really acidic because it has a lot of hydrogens in it. Now what happens is there's a protein right here, it's called ATP synthase. And if you ever go farther into biology, you'll learn this is a crazy important molecule that your body uses all the time and has to be everywhere. It's really cool and does a lot of awesome things. But for here, ATP synthase basically uses acid. It uses the hydrogen atom inside of this space here to effectively act like a pump. And what happens is that as a hydrogen moves through this ATP synthase, it turns an ADP molecule, adenosine diphosphate, and turns it into ATP. So in this process, you can see that a water molecule, which is why plants need water, is split apart into hydrogen and oxygen, and then an electron moves through the system using light at various steps along the way, and it actually turns that electron molecule, combines with NADP and a hydrogen to create NADPH, And the hydrogen molecule that was put in this little in the thylakoid space inside of the thylakoid, which are these little small pancake-looking things here, these hydrogens that escape out of the ATP synthase protein right here and create ATP. So the light-dependent reactions create two molecules. It creates NADPH and it creates ATP. Now there are some other things you're going to see around here, like the word activated electron. That's just a fancy way of saying it's the electron that's going to transfer through all these proteins right here. You have the electron carriers. These electron carriers, their one job is they are proteins that are capable of moving electrons. So essentially what's happening is electrons are traveling through these electron carriers. They're kind of like just handoff guys. They ground the electron and they pass it to the next guy. So that these guys are pretty simple in what they do. And this is the electron transport chain for photosynthesis. It's important to understand there are two electron transport chains, which I haven't talked about yet, but I'm going to give you a heads up right now. This is electron transport for photosynthesis. There's also one that takes place in cellular respiration as well, but I'll talk about that in the next lesson. So, stage 2. As I showed you guys beforehand, what happens is that the electron transport system that creates NADPH and ATP, both of these molecules, they are energy molecules. You can use them as petrol in your body if you wanted to. But the problem is, though, they're very unstable. They break apart really easily. So they're really, really bad for storage. So the plant doesn't want to store these molecules because it's going to lose a lot of them just because of bad luck, or they'll bump into each other and break apart. These are very, very weak molecules. So what happens is the plant has to have a solution to that problem. And the plant solves that problem by creating a molecule that it can store energy for a long time. That molecule that can store energy for a long time is called sugar. It's glucose. So the plant wants to turn NADPH and ATP into a long-term storage. It turns that molecule into sugar. Now, this is where the second step comes in, the light independent reactions, because you don't have to have light anymore. The step of turning these two molecules here into sugar is basically the light independent reactions. Now, this is called the Calvin cycle. The Calvin cycle just takes carbon dioxide out of the atmosphere. It turns into a molecule called C3P, which you'll talk about in a little bit in the video I'm going to show you guys. And by using ATP and NADPH, essentially it turns this C3P into a sugar molecule, which can be stored inside the vacuole to be used later. That's all the Calvin cycle does. So this right here is a good little diagram of the Calvin cycle. What happens is you have 6 carbon dioxide molecules come in. So you have your 6 carbon molecules here. It's going to combine with a molecule called PGA3. Don't worry, you don't have to know these fancy terms because I don't remember them half the time. This is people who are really into biology on the cell side. What happens is that the cell invests the chloroplast invests 12 ATP and 12 NADPH, which were created in the electron transport chain, which creates 12 of the G3P molecules, which then can be used to create through Rubisco, which is just it's an enzyme, and that enzyme Rubisco is going to turn on these molecules with the help of 6 ATP into this 6 ribose biphosphate, these 5 carbon molecules. It'll then take another carbon and then repeat the process. Now if you look right here, 2 of these G3P molecules right here were broken off, and these will be transported from the chloroplast to make glucose and fructose and starch. These are all just basically sugars. So essentially what happens is you have this cycle where the carbons are being recycled through the whole process, where ATP and NADPH are being put into the cycle to help create sugars. Now, the video I'm going to show you guys here in a minute does a much better job of explaining this than I can, because it's really hard to see what's going on just by looking at this graph. But the video I'm going to show you guys will definitely make this thing a lot clearer. So, the Calvin cycle, what I want you to know: it takes carbon dioxide from the air, it takes that carbon dioxide, it invests ATP and NADPH, and then obviously more ATP here, and that investment of these molecules creates sugar. So, there are some alternative ways of doing things. There's two ways of doing things that are different from the cycles I told you about here. The first one's called a C4 pathway. A C4 pathway basically is just a way to save water. Some plants they make a molecule that has 3 carbons rather than the usual type, it has 4 carbons rather than the usual 3 carbons, and essentially it's a different way of doing things. It saves water. You don't have to know much about this. Just know that the C4 pathway is an alternative way of doing the Calvin cycle, that rather than having 3 molecules right here, you're going to have 4. And that plants in dry climates, like here around Riyadh, are the ones that are more likely to do the C4 pathway. The last one is called a CAM plant. Essentially what a CAM plant does is that it doesn't use any of the light independent reactions during the daytime, because if it has to open up its stoma in order to do that, which are little tiny holes in the plant, and unfortunately water can escape from those small holes. And these plants, what they do is they alternate their light independent reactions and their light dependent reactions depending upon the time of day. And the idea behind that is, as long as the less openings you have in this plant, the less water can escape. So again, a lot of desert plants do this as well. So they will save water by only letting in carbon dioxide at night. That's all a CAM plant does. They do everything else is the exact same. They just alternate what time of day they run the first step and the second step of photosynthesis. Okay, I'm now going to show you guys a TED Ed video that does a great job of explaining how the Calvin cycle works. So I'm going to go ahead and splice that into this video. So if things work well, you should see the video pop up here in a minute.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3 phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just one carbon that gets soldered to one RUBB chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
All right, that was the video. I hope you guys enjoyed it and hope things make a lot more sense now than they did beforehand. Feel free to watch that video again to help you understand. If not, I can help by helping you in class. Just make sure to raise your hand. Okay, lesson in review. So there was a couple things about this lesson. First off, photosynthesis. You take CO2, water, and sunlight, and you turn it into sugar. That's all it is. Now, photosynthesis has 2 different steps. There's light-dependent and light-independent reactions. The light-dependent reactions are used to make ATP and NADPH, and the light-independent reactions are used to make are used to take ATP and NADPH and turn them into sugar. The last part is the Calvin cycle. That's just a fancy way of talking about the light independent reactions, because all the Calvin cycle does is it takes ATP and NADPH and turns it into sugar. And that's it. Oh, sorry, one more thing. There's electron transport chain. That's the idea of using light and water to create NADPH and ATP. This is part of the light-dependent reactions. And the last part here, guys, is make sure you know what the parts of the chloroplast are. I believe if things work out the way they're supposed to, this will probably be part of your practical exam. So you will want to know what the various parts of a chloroplast are. Okay, for the activity today, guys, I'm done talking. You want to grab the photosynthesis activity. It should be in the folder I gave you. And you want to complete this activity and put it into your Evernote portfolio. And thank you for watching, guys. Sorry for the long link to the video. If you have any questions, as always, raise your hand in class, and I will be here to help you. Thank you very much. Have a good day.